Sounds like everybody's tired. It's quiet in here. Happy Friday, anyway. So today we're going to dive a little bit deeper into the control of breathing. We're going to look at some factors that can affect both the rate and depth of breathing. Okay. So this is the slide where we left off on Wednesday. And if you remember, I pointed out that there are 3 different respiratory centers. And I finally got this text. For some reason, somehow these got overlapped. I got that fixed now. The 3 respiratory centers that affect breathing are the ventral respiratory group, which sends out the basic pattern of breathing. There's also a pontine respiratory group that's located up in the pons. Pontine respiratory group can either slow down or accelerate breathing. But it doesn't stimulate the ventral respiratory group directly. It does so through the dorsal respiratory group. So the signals pass from the pontine respiratory group down to this dorsal respiratory group, which is the larger of the 2 down here. And then the dorsal respiratory group stimulates the ventral respiratory group, and then that modifies the rate of breathing. Okay? So you might say that the dorsal respiratory group is just sort of a relay center. It relays information to the ventral respiratory group from other parts of the brain. Okay. Now, in addition, there are descending pathways from higher brain centers, which is what allows us to voluntarily change our pattern of breathing. Like if we wanted to hold our breath, we can do that. Or if we want to voluntarily hyperventilate, I don't want to do that for too long, you'll start getting dizzy because you'll blow off too much carbon dioxide. But you can do that because there are descending pathways from higher brain centers down to the respiratory centers. In addition, as we're going to see today, there are chemoreceptors located in 2 different areas of the body. There's central chemoreceptors, which are located in just inside the wall of the 4th ventricle down here. And there's also peripheral chemoreceptors. The peripheral chemoreceptors are located in the carotid bodies, which is right where the external and internal carotid artery come off of the common carotid artery. These chemoreceptors monitor the carbon dioxide in the blood. And if CO2 builds up, then that stimulates the chemoreceptors. Chemoreceptors send that information to ultimately to the ventral respiratory group, but it goes through the dorsal respiratory group first. And then that will stimulate more breathing. Okay? That way you blow off more CO2 and that restores homeostasis. Now, there's a little bit more to the story than that, but because the chemoreceptors actually monitor CO2 indirectly. They do so by monitoring the pH of the blood. But pH is affected in the blood primarily by CO2 levels. And so as the CO2 levels go up, your pH goes down. As your CO2 levels go down, your pH goes up, becomes more basic. Okay, so we're going to look today, I'm going to start by looking at how that happens. All right, so as I said, there are central and peripheral chemoreceptors. The central chemoreceptors monitor the pH levels of the cerebral spinal fluid in the brainstem. And the pH of the cerebral spinal fluid is influenced primarily via CO2 levels of the blood. Now, hydrogen ions, which is what affects pH, are also produced from other things besides CO2. Hydrogen ions can also be produced by metabolic acids, like lactic acid. However, lactic acid cannot go across the blood-brain barrier, so it can't get into the cerebral spinal fluid. So all of the hydrogen ions in the cerebral spinal fluid are produced when carbon dioxide dissolves in the cerebral spinal. Lactic acid can't go across the blood-brain barrier, so it can't get into the cerebral spinal fluid, but CO2 can. CO2 can go right through the blood-brain barrier. It's permeable through that. And then once it gets into the cerebral spinal fluid, carbon dioxide combines with water with the aid of an enzyme that's in the cerebral spinal fluid called carbonic anhydrase. And that converts the CO2 to carbonic acid, which is a weak acid. Carbonic acid almost immediately disassociates into bicarbonate and hydrogen ions. And it's those hydrogen ions then that can stimulate these central chemoreceptors. So, the central chemoreceptors respond indirectly to CO2 levels via the hydrogen ions that the CO2 produces when it combines with water. And then the chemoreceptors can send signals to the respiratory center, modifying the pattern of breathing. Okay? So if CO2 levels go up, then that causes an increase in ventilation. All right? Now, in addition, there's also peripheral chemoreceptors. Peripheral chemoreceptors are located in the same general area as the baroreceptors, which monitor blood pressure. Okay? There's also structures located in that same area called the carotid bodies. And those carotid bodies contain cells that primarily monitor the hydrogen ion levels in the blood. Not the cerebral spinal fluid, but there's blood in those blood vessels. And in addition, the peripheral chemoreceptors can also be stimulated by relatively drastic drops in oxygen levels of the blood. But the peripheral chemoreceptors, along with the central chemoreceptors, respond mainly to CO2 levels by way of the hydrogen ions they produce. The central chemoreceptors doesn't respond to oxygen changes at all. They're just not sensitive to that. But the peripheral chemoreceptors will be stimulated if there's very large drops in CO2, or excuse me, of oxygen levels. Okay. But primarily they're affected by, they can be affected by rather small fluctuations in the pH of the blood, which is affected again primarily by CO2 levels. But they can also be stimulated by large drops in oxygen. Small changes in the oxygen levels of blood don't affect breathing at all. It has to drop quite low before it will stimulate the peripheral chemoreceptors. Now, since the blood in the circulatory system is not protected by any kind of barrier like the blood-brain barrier, there are other compounds in the blood that can produce hydrogen ions, which lowers the pH of the blood. So there are metabolic acids like lactic acid and citric acid that's produced in the Krebs cycle, as well as other metabolic acids. And so hydrogen ions produced by lactic acid can also affect the pH and therefore stimulate the peripheral chemoreceptors. Okay? So if a person, let's say, takes a whole bunch of antacids. Antacids are basic. Bases neutralize acids. That's going to raise the pH of your blood. That's going to actually affect the chemoreceptors because the chemoreceptors can't tell where the hydrogen ions come from. All they know is that there's hydrogen ions there. So it doesn't matter the source of the hydrogen ions for the peripheral chemoreceptors, but the central chemoreceptors are only affected by changes in pH associated with CO2 levels. Okay? So, peripheral chemoreceptors respond indirectly to CO2 levels via the hydrogen ions they produce, metabolic acids like lactic acid, and very large decreases in oxygen saturation of the blood. Okay. But you actually have to hold your breath for quite a while before your oxygen levels would fall low enough to stimulate those peripheral chemoreceptors. Okay. So peripheral chemoreceptors, again, are located in the aortic arch and also the carotid sinuses. So this is just a diagram of the heart and the large arteries that supply the upper part of the body. So there are aortic bodies down here which contain peripheral chemoreceptors in the aortic arch. And also up here, right in this area, that's called the carotid sinus. There's carotid bodies up there that contain chemoreceptors. And so these two locations of the peripheral chemoreceptors, that's in addition to the central chemoreceptors that are located in the brainstem, very, very close to the cerebrospinal fluid. Okay, as I said earlier, it is possible to have voluntary control over breathing, and those signals originate from the cerebral cortex of the frontal lobes. Those neurons in your frontal lobes can initiate action potentials which travel down through what they call cortical spinal tracts in the brain to respiratory neurons in the spinal cord, bypassing the brainstem. But there is a limit to voluntary control, because there's actually a breaking point where a person is forced to breathe. And that happens when CO2 levels rise to a point that the autonomic control overrides the voluntary control of breathing. So you can't suffocate yourself by holding your breath. Sooner or later, that involuntary control overrides your central control of breathing. So this is just kind of a flow chart that summarizes the factors that affect breathing. So keep in mind, and this is going to be important next week when we're going to do a lab that has to do with the control of breathing. Carbon dioxide is the main factor. Levels of carbon dioxide, main factor that control breathing. Okay? Because when you're at rest, oxygen levels never fall far enough that it affects the peripheral chemoreceptors. And so it's CO2 that's controlling the rate of breathing most of the time. So CO2 can affect both the medullary chemoreceptors, which are also known as the central chemoreceptors in the brain, as well as the peripheral chemoreceptors, which are located in the carotid sinuses and in the aortic arch. Those chemoreceptors are stimulated by CO2 by way of hydrogen ion production, because CO2 will dissolve in the plasma just like it does in the cerebrospinal fluid, and it combines with water to form carbonic acid, which gives off hydrogen ions. So it's indirectly monitoring CO2. To be technically correct on this diagram, they should put a hydrogen ion up there instead of CO2, because it's actually the hydrogen ions that are directly stimulating the chemoreceptors. Okay, then over here on the top right, you can see it says oxygen and pH. Large drops in oxygen can also stimulate only the peripheral chemoreceptors. It doesn't affect the central chemoreceptors. And changes in pH caused by metabolic acids like lactic acid, that also can affect the peripheral chemoreceptors. Okay, and so that's those signals from the chemoreceptors are carried to the medulla oblongata and pons by afferent sensory neurons, and then that's where the respiratory center is located. That's where those three respiratory centers, the ventral respiratory group, the dorsal respiratory group, and the pontine respiratory group. And so, in response to the input from the chemoreceptors, that causes a change in the length of the inspiratory bursts of neurons. So, if that inspiratory burst is shortened, that's going to accelerate breathing, because the shorter the inspiratory period, the more inspirations you can have per minute. If the respiratory burst of action potentials is prolonged, that's going to slow down breathing, but it's going to increase the depth of breathing. Okay, because in general, there's an inverse relationship between the depth of breathing and the rate of breathing. As depth increases, the rate decreases. And the reason for that is when depth increases, it takes longer for inspiration to occur. If inspiration takes a longer time period, you're gonna have fewer respiratory cycles per minute. So that's going to slow down your breathing. Okay. Conversely, the shorter the inspiratory burst, the faster the respiration, because you can fit in more respiratory cycles per minute. Okay. In addition, it's also possible to modify your basic breathing pattern via voluntary control from the cerebral cortex and the frontal lobes, as well as emotions that are produced by the limbic system, and that's deeper in the cerebrum. Those actions are carried out by higher brain centers in the limbic system. There are inputs from that into the respiratory centers, and so that can modify the signals that travel to all of these respiratory muscles by the voluntary somatic neurons. Okay? Because respiratory muscles are skeletal muscles, which are voluntary, so those are stimulated by somatic motor units. Somatic motor division is the division that stimulates the voluntary muscles. Okay? During quiet breathing at rest, your respiratory stimulates your diaphragm as well as the external intercostals. During forced inspiration, expiration, then there are some additional muscles that are stimulated. For forced expiration, your internal intercostals and your abdominal muscles are also stimulated. Okay. Any questions? Okay. All right. Now, in order to understand ventilation or breathing, we need to know a little bit about the behavior of gases, because air is basically a mixture of gases. And so the first thing we need to look at is a relationship between pressure, resistance to airflow, and the flow of air itself. And so airflow is governed by the same principles that govern blood flow. So in other words, air always flows from an area of higher pressure to lower pressure. Same thing as blood. So the flow of fluid, like blood, is directly proportional to the pressure differences between 2 points. So you have to have a pressure differential or you'll have no airflow. In addition, the flow of a fluid, whether it be blood or whether it be air, is inversely proportional to the resistance resistance is what prevents airflow, okay? So the flow of air is directly proportional to the difference in pressure between 2 points divided by the resistance to that flow. And that resistance to the flow of air is primarily the friction between the air molecules and the inside of the airways. When those air molecules collide with the inside of the conducting passages of the respiratory system, that creates a friction that slows down the movement of those air molecules. All right, now, there's also some gas laws that you may have heard about when you were in chemistry. And these also play a role in airflow. One of these is called Boyle's Law. Boyle's Law states at a constant temperature, the pressure of a given quantity of gas is inversely proportional to the volume. Okay? This is kind of common sense, I think. So as volume of a space increases, like the volume of the inside of your thoracic cavity, as that increases, that causes a drop in pressure. When volume decreases, it causes just the opposite. It causes air pressure to increase. Okay, so there's an inverse relationship between volume and pressure. And so when the volume of your thoracic cavity expands during inspiration, the pressure is going to fall. When that pressure inside your chest falls below that of atmospheric pressure, then air is going to flow from the atmosphere into your lungs, and that causes your lungs to fill with air. If lung volume decrease, then the pressure inside your lungs is going to increase. And so that's going to cause air to move from your lungs out to the outside of your body. Okay, and that's going to cause expiration. So this is just kind of a summary for how breathing occurs. It's just a very short, concise summary, actually. So we start out up here at the top. So at rest, the diaphragm is dome-shaped, and it actually kind of pushes up into the thoracic cavity. And at rest, atmospheric pressure and intrapulmonary pressure. Intrapulmonary pressure is the pressure inside your lungs. At rest, that pressure is the same between your lungs and the atmosphere. So there's no movement of air in or out of your lungs. But during inspiration, when your diaphragm and external intercostals are stimulated, that causes your rib cage to move upward and also slightly forward, kind of like lifting up the handle of a pail. And at the same time, your diaphragm flattens out and drops down. So both of those are going to increase the volume. When the volume of your lungs increases, then the pressure is going to drop. And when the pressure drops below that of the atmosphere, then air is going to go from the atmosphere, which is higher pressure, to an area of lower pressure in your lungs. So that's going to cause your lungs to fill with air. And then gas exchange can occur. Okay, during expiration, at least during quiet expiration, those signals that are stimulating your diaphragm and your external intercostals, they stop being produced. And so your rib cage just undergoes elastic recoil, returns to its resting position, and your diaphragm moves up because this resting shape is more dome-shaped. That reduces the volume, increases the pressure inside your lungs, and so the air goes from the area of higher pressure to lower pressure, and the air escapes from your body back out into the atmosphere. And then the cycle starts all over again. So that's basically the respiratory cycle in a nutshell. Okay. All right, now there's also another gas law that has a smaller effect on breathing, and that's Charles' law. Charles' law states that the given volume of a gas is directly proportional to its absolute temperature. So on a cool day, 16 degrees Celsius or 60 degrees Fahrenheit inhaled air will increase its temperature by 21 degrees Celsius as it enters the body. So inhaled air is warm to 37 degrees by the time it reaches the alveoli. When the temperature of a gas increases, that causes an increase in pressure. So that's another factor that contributes to the inflation of your lungs. When you breathe in cool air, it's warmed as it goes past the mucous membranes in your nasal cavity, and it's warmed as it goes through the conducting part of your respiratory system. When that air warms up, that increases its pressure. And so that helps to inflate the lungs, along with those pressure changes that were a result of Boyle's law. So an inhaled volume of 500 milliliters will expand to 536 milliliters, and that helps to inflate your lungs. But the most important factor is the pressure differences caused by Boyle's law, which describes the relationship between volume and pressure changes. Okay. Now, another factor that affects airflow is resistance. The greater the resistance, the slower the flow. And there's three factors that affect airway resistance. The most important one is the diameter of the bronchioles. The bronchioles are some of the smallest conducting airways of the respiratory system. They're the smaller branches of the tertiary bronchi. And the bronchioles have a layer of smooth muscle in their walls. They have fairly thick walls, actually, which is why there's no gas exchange occurring there. And those smooth muscles can be stimulated by the autonomic nervous system. So when the sympathetic system stimulates the smooth muscles in the bronchioles, that causes bronchodilation, which increases the diameter of the bronchiole. So bronchodilation, and that's going to reduce resistance to airflow. It's going to make air flow in more easily. So sympathetic stimulation, as well as epinephrine produced by the adrenal medulla, both can cause bronchodilation. Okay. So when does sympathetic stimulation occur? Under what kind of situations does that occur? Think about under what kind of situations do you need more airflow in your lungs?
Exercise?
Exercise, yeah. Yeah, during exercise, there's an increase in sympathetic stimulation. What else? What other kind of situations would you need an increase in airflow that would cause the sympathetic division to stimulate your lungs? What's another name for the sympathetic division? Fight or flight division. Okay. So if you get scared, okay, so let's say you're out at night after dark all by yourself, and all of a sudden a big group of people comes up behind you and they're kind of rowdy. you'd probably get a little bit frightened from that, okay? And so that would result in activation of your sympathetic division. That would stimulate the smooth muscle in your bronchioles, cause bronchodilation, which would increase airflow. Now, the opposite of that is bronchoconstriction. That causes a decrease in the diameter of the bronchus or bronchiole. There's a number of factors that can cause bronchoconstriction. Histamine, which is produced as a result of inflammation. So like if you are allergic to dust and you inhale a lot of dust, it's going to cause histamine to be released in your lungs. That's going to cause bronchoconstriction. That's why people that have asthma, when they inhale something they're allergic to, makes it harder for them to breathe. They have an asthma attack. Another factor is parasympathetic stimulation. Parasympathetic division has the opposite effect of the sympathetic division. So that's going to cause bronchoconstriction. So the parasympathetic nerves might stimulate your bronchioles when you sit down and you start to rest, because you don't need as much airflow in your lungs when you're resting. Cold air can cause bronchoconstriction. That can stimulate the smooth muscles. And as I said, chemical irritants. So, if you breathe in acid fumes or something, that can cause bronchoconstriction. That's sort of a protective reflex. All right, then a second factor that affects airflow is what's called pulmonary compliance. Now, the pulmonary compliance is defined as the ease with which the lungs can expand. Okay? So if you have high compliance, that means that the lungs can expand very easily. Normally, your lungs do have good compliance. Okay? However, some disorders can reduce pulmonary compliance. One example is if someone has COPD, that can, actually a better example would be cystic fibrosis. Someone has cystic fibrosis, the airways and the lungs produce too much mucus, and that can cause damage to the lungs, and it can cause scar tissue to form in lungs. If scar tissue is formed by lungs from something like long-term smoking, or if someone has a disorder like cystic fibrosis, that can make it more difficult for the lungs to expand. And so that will reduce pulmonary compliance. Okay? That will make it harder for a person to breathe. You have to work a lot harder. You have to expand your chest more. You have to increase the activity of your respiratory muscles in order to take in the same amount of air as a normal person. Okay. So any diseases that cause a breakdown of the alveoli and the formation of scar tissue, that can decrease pulmonary compliance, make it more difficult to breathe. And then a third factor is the surface tension of that thin layer of fluid that lines the alveoli. All respiratory surfaces are covered with fluid, because that's the only way for oxygen to get through the walls of the alveoli is to dissolve in water. Oxygen has to be able to dissolve in water. It doesn't dissolve very well in just the cell membrane. And so, because there is water lining the inside of those respiratory surfaces, the alveoli would have a tendency to collapse. And once they collapse, it would be hard to inflate them again. And so to prevent that from happening, our lungs have type 2 alveolar cells, which produce a phospholipid called surfactant. And that surfactant reduces the surface tension of water. Just sort of like putting soap in water. That's going to reduce the surface tension. And as I said, the other day, that sometimes happens, or surfactant isn't produced sometimes with premature infants. They're born prematurely because surfactant isn't produced until just right before delivery. So if a baby's born prematurely, it may not have yet produced surfactant. That can cause the alveoli in the baby's lungs to collapse, and it makes it hard for them to breathe. And so that can result in what's called infant respiratory distress syndrome. And because of that, sometimes babies that are born prematurely, without, at sudden notice, sometimes they have to be placed on respirators to help them breathe. Because without surfactant, their alveoli collapse because of the surface tension of water. This is also known as hyaline membrane disease, because supposedly their lungs have a hyaline, or sort of a glassy appearance. All right. So, only that air that enters the alveoli is available for gas exchange. But not all the air that we inhale gets into the alveoli. About 150 milliliters of the air that we breathe in just fills the conducting division of the respiratory system. So those are the larger airways that have thicker walls, and there's no gas exchange that can take place there. So that air that stays in the conducting division of the respiratory system is called, that area, that volume, is called the anatomical dead airspace, because no gas exchange can occur. It's just dead air. And that amounts to about 150 milliliters. And so in some pulmonary diseases, like COPD, the alveoli may be unable to exchange gases because they either lack blood flow or the respiratory membrane has been thickened by fibrosis, which is the formation of scar tissue. That essentially increases the effective anatomical dead airspace. And so it decreases the amount of air, gas that can be exchanged in the lungs. All right. So, in summary, air enters and leaves the lungs as a result of pressure changes as well as temperature changes. Those pressure changes cause changes in the volume, or as a result of the changes in the volume due to contraction of respiratory muscles. The temperature change that occurs in the air that's inhaled also contributes a little bit to the expansion of the lungs. You need to remember that quiet inspiration is due to contraction primarily of the diaphragm and external intercostals. External intercostals are the outer layer, superficial layer of muscle between your ribs. Expiration, however, at least quiet expiration, is mainly a passive process. It just occurs when those inspiratory muscles relax. And there's an elastic recoil of your chest, and your diaphragm moves back up into its normal dome-shaped position. And then the basic rhythm of breathing is produced by the ventral respiratory group, which is located in the medulla oblongata. And those signals, though, are modified by the pontine respiratory group, as well as input from the peripheral and central chemoreceptors, as well as input from higher brain centers sometimes, not all the time, but sometimes. If you voluntarily decide to change your breathing pattern, then signals descend from your higher brain centers to your respiratory center. And also, sometimes signals from your limbic system can change the breathing pattern. That's why people that have something really bad happen to them and they start crying, that changes your breathing. They sob and they take deeper breaths. It's because of input from that limbic system. All right, any questions? Okay, so let's go back to this diagram. I want to ask you a question. Okay. First diagram we looked at. There we go. Okay, I'm going to ask you a question. So let's say something happens, like a person gets in an accident, and the dorsal respiratory group gets completely destroyed, as well as the pontine respiratory group. Would that person be able to breathe? So the dorsal respiratory group is damaged, pontine respiratory group is damaged, but the ventral respiratory group is still intact. So would that person be able to breathe? I see you nodding your head up and down, yes. Okay, that is correct. Yes, you still would be able to breathe. You can actually breathe without your dorsal respiratory group and your pontine respiratory group. And that's because as long as your ventral respiratory group is intact, it's going to keep sending out signals, bursts of action potential, which would cause you to breathe. So you'd still be able to breathe. But there'd be one problem, though. What would be the problem? Would your breathing rate be able to change if the only respiratory center that was functioning was the ventral respiratory group? No, it wouldn't. Or the breathing pattern would stay the same all the time, okay? Because without input from the pontine respiratory group, well, actually, no, I'll take that back. If your ventral respiratory group was intact, you would still have input from the central chemoreceptors. So if the pH of your cerebral spinal fluid decreased, that would stimulate your ventral respiratory group. That would allow your breathing to accelerate a little bit, but probably not as much as if your pontine respiratory group was still intact, because that would be able to send signals down to the dorsal respiratory group, followed by the ventral respiratory group to modify breathing. So you might have a little bit of an ability to modify your breathing rate, but not very much. So if that happened, and if a person was in that situation, and you were to start exercising strenuously, you might not be able to increase your respiratory rate enough to meet your body's demands during exercise, because your pontine respiratory group wouldn't be able to stimulate your respiratory centers in your medulla. Okay. So yeah, you'd still be able to stay alive, but you wouldn't be able to regulate your respiratory rate as much. Okay. Well, that's all I have for today, so this would be a good place for us to stop.